My name is Jonny Wray. I'm from e-therapeutics, and I wanted to give you an overview today of some of the work we've been doing at our company and how we're utilizing in-memory computing in that effort. So, quick overview, who we are, what we do. We're actually a small drug discovery company we're actually based in Oxford, not far from here. And I guess what we do differently, our key difference is that all of our early stage discovery is done in silico using computational modeling, hence my involvement today. Given the audience, I thought I would sort of give a very quick overview to the drug discovery and development process and where some of the challenges are currently. So this diagram is a pictorial representation of the whole drug discovery and development process. So you can see there's sort of three core chunks to it. There's a discovery phase, a development or clinical phase, and then a post-discovery phase. e-therapeutics, my company, works in this preclinical early stage drug discovery phase. And what our core goal scientifically is to try and identify potential compounds that can be then made into drugs that will treat disease. A couple of points on top of that I wanted to bring out from this diagram, and that one is the time frames. If you look across the bottom, it gives you an idea of how long the drug discovery process takes. Basically anywhere from 10 to 15, 20 years from initial idea through an approval. That's rather a slow feedback loop. A lot of that affects what we do. The other thing I wanted to bring out is just the attrition rate. And I actually think some of these numbers are quite optimistic on here. But they're saying, of all of the 10,000, I would actually say it's maybe an order of two of magnitude higher than that, 100,000 million compounds tested. At the beginning, one might make it to final approval. This next slide, I guess, gets at the maybe some of the things that came up in the keynote this morning on the digital transformation that's actually starting to happen in our industry as well. I think there's some evidence that our drug discovery industry is a bit behind in that adoption of digital technologies, but it certainly is happening. And it's certainly happening at small, innovative companies like ours, maybe not so much in the big drug companies, but they are certainly catching up. So, graph on the left-hand side, this is a graph of productivity. I believe it's the number of drugs discovered over time for, per million or per $1 billion. I can't read the axis at the moment. It's an exponential scale. It's a log axis on the y-axis. And actually, the guy who wrote this paper called it Eroom's law. That's Moore's law in reverse. Basically, the productivity of our industry has been massively declining over the last few years. And this is on top of all of the advances in the underlying technology of examining biological systems, all of the stuff that's come along post-genome sequence, etc. Huge number of new technologies in the lab, productivity still massively decreasing. Middle graph, I'll just pay attention to the black bar on the right. That's the cost to bring one drug to market. It's $2.5 billion currently. And that was done a few years ago now, so it's going up. That's a lot of money. And I guess the 2 points I wanted to make or the 1 point I wanted to make from this is that our industry is ripe for innovation. It's ripe for new technologies, trying to improve the productivity at multiple different stages of the drug discovery process. And this is certainly starting to happen in our industry now. People are applying things all the way through making clinical trials better, potentially, through to early stage optimizing compounds in a more efficient way. That's actually proven to be one of the more successful applications of artificial intelligence and active learning in our field, is how to optimize chemical efficacy. And where we operate is motivated by the graph on the right-hand side. And this was just one figure that I picked out of a paper from some people at AstraZeneca, one of the big UK drug companies, who did a paper on analyzing the reasons for failure. And it's a big paper, lots of reasons. What this one's highlighting is that a large percentage of the late-stage failures, so that's failures that get to the last stages of clinical trials, so that means a hell of a lot of that $2.5 billion has already been spent on them. A lot of those failures are due to the fact that the right biology is not being perturbed. Something went wrong right at the beginning. All the chemical optimization, et cetera, that's happened in the meantime has been optimized against the wrong thing. And that's where we operate. We're trying to use informatics approaches that I'll go through to try and actually come up with ways of investigating complex biology more efficiently and hopefully coming up with drugs that reduce this late-stage failure and therefore lead to better productivity in the industry. So enough of the motivation from a business point of view. How do we address this? Bit of biology. There's a field, academic field that's been called network biology in the last 10, 15 years that's sort of come up, and this is basically a field of study around consideration of the cell, a cell as a network, as a social network, but rather than between people, between molecules within the cell. What I've illustrated on here, there are a number of different types of interactions that make up networks within the cell. Protein-protein interactions, that's literally proteins talking to each other, physically affecting each other. Metabolic networks, these are the molecules involved in energy processing within our cell. Signal transduction networks, these are the things that are responsible for taking a signal from outside of the cell, getting it into the cell and where it needs to go to. And gene regulation networks, these are the networks that underlie what regulates a gene. And what I mean by that is, every cell in our body has the same genes in it. What makes cells different from each other, liver cell different from a heart cell, etc., is what genes are regulated, which ones are actually active in that cell. That's controlled by networks of things, not individual elements. The cell itself is actually an amalgamation of all of these networks. The fact that they're different is just due to experimental techniques, really. But one thing that comes out of this is key, is that biological function at a cellular level can't be ascribed to any one molecule. It arises as a function of the network of a bunch of things talking to each other. And this includes disease function. So a couple of experimental papers that sort of outline this. First is this concept of dysregulated network modules. So there's a reasonable amount of evidence that in cancer, one of the things that can be driving cancer is, I mentioned the gene regulation networks. Quite often what a cancer cell will do is turn on regulation patterns that are usually active during development. Cancer, we know cells carry on proliferating, they don't stop dividing. That's what happens in development. It gets turned off due to gene regulation changes. Cancer turns these things back on, basically. You end up with dysregulation. But it's due to a network effect. It's not due to single gene effects. Another interesting one, actually, is the idea of pathological interaction. So this is an example I took from a paper on Huntington's disease. Huntington's disease is actually due to one genetic problem. I was going to say mutation. It's not actually mutation, but it doesn't really matter. There's one problem in one gene in the disease. It gives rise to the Huntington's behavioral changes, phenotypic changes. However, the field has changed from looking at, okay, how can we change, what's going on with that one gene, to actually looking at what happens after that gene changes, and effectively the cellular networks rewire, and you end up with a pathological network. So the actual disease, how it manifests itself, isn't anything to do with the gene that changed it, that triggered it. It's to do with the network that it ended up with. Hence, the thing you should be targeting in drug discovery is the network, not the individual genes. So it's these underlying concepts that disease behavior, arises from a network that drives what we do. And this is just a diagram to illustrate this. That complexity on a biological level basically exists in an increasing hierarchy from a gene, DNA, all the way up to behavior, arising from more and more complex networks. Now, the issue for the practical application to drug discovery is that, okay, so we're trying to change behavior, trying to change disease behavior. I've just told you, hopefully successfully, that arises from an interaction of multiple molecules, but a drug interacts at a molecular level. So there's a gap there that needs to be bridged. And going after networks, using networks as your key thing that you're trying to analyze, in our case, or perturb, bridges that gap. So that's all very well in theory. How do we actually turn that into a practical engineering solution in a drug discovery context? So this is what we do as a company. So the first stage in our process, if we're going after a new indication, is to, sorry, I just want my timer, is to look for gaps in available treatment for a disease. No point in going after a disease that's well treated, that's not amenable to our approach, etc. Every drug company would do that. It's not particularly novel to us. The next stages that are illustrated in pink are probably novel to us. The first is what we call the identification of intervention strategies. So this is looking at a disease, looking at its pathological progression, what point does the patient go to the doctor, and trying to identify at that point in time what mechanisms are operating that we can intervene on potentially at a system level, so there's all system level concepts, to have a positive effect on the disease. Now, we're dealing with biological data at this time. Biological data is notoriously messy, biased, noisy, etc. So we don't tend to hang a hat on one strategy here. For a particular project, we'll usually come up with multiple strategies representing different sets of assumptions, etc. Once we've identified what we want to do, we then move into an in-silico phase. And these are, I'll delve into the technologies underlying these in coming slides. But to give you a sort of quick overview, there's three distinct stages. The first is model construction. So we build in silico network-based models of the disease processes we want to disrupt with a drug. These are constructed from data integration, and we end up again with multiple networks for a particular problem, encompassing different sets of data, different assumptions, etc. We then go into a network analysis phase. We have a number of techniques we use, but they all try and get at the same problem, which is, given a network that we believe underlies disease, how do I disrupt it? How do I stop these things talking to each other in the most effective way? Biological networks are robust to perturbations in general, much like the internet and other networks are. It's actually thought that that's what underlies biological robustness. If it didn't, none of us would make it to be able to procreate, which is what drives evolution. So there's an inherent biological robustness. Individual molecules in the cell aren't robust. The networks that they interact with are robust. The final stage, once we've analyzed the networks and we've come up with a sort of collection of optimal, idealized ways of disrupting those networks, we go into a compound mapping phase, which asks the question, okay, what potential compounds could give rise to that pattern that our analysis identified? I mentioned the issues with noise, biased data. This is that consideration has driven the process and the design of the process. So the output of our process, our analytics, isn't small numbers of predictions, and we're not trying to make small numbers of accurate predictions on small number of molecules. It can't be done at the moment, we don't believe. We take a statistical approach where the output of our system is a list of compounds we believe are going to be enriched in actives over what you would expect by chance. And then we take them into the lab. So we take a list of compounds and then we put them through what's known as a phenotypic screen. So in our language, screen is just a test in the lab, and a phenotypic means it's a functional test. So does it kill cancer cells, for example, would be a phenotypic screen. So you're trying to say, do these compounds do what we expect them to do? And those tests that we do at stage 5 represent the mechanisms that we identified in step 1 that we believe should be important. Once we get actives, we call hits in our field, out of those screens, they then optimize towards a lead. A drug has a number of properties other than it does what you want it to do to cancer cells, kills them, that it needs to have. It needs to get to the tissue, it's targeting, it needs to stay around long enough to have the effect, it needs to not be metabolized very quickly or not at all, because then it would be toxic, etc. So there's a whole host of other properties that make a good drug, and these are generally brought in a chemical lab-based optimization phase. And I mentioned earlier that's one of the areas, not that we do, but other companies do, that have applied sort of active learning approaches quite successfully in. There's a couple of videos here. Rather than sort of try and describe what we're trying to do, I thought I'd illustrate it. So these are networks that actually came from one of our discovery projects. The red balls are proteins. The blue lines are connections between those proteins, and they make up a network. This network underlies a disease process that we want to disrupt with a network, with a drug. The point that slide is supposed to make is that if you make random perturbations to that network, not much happens to the structure of the network. It's robust. I mentioned that before. If you make targeted perturbations using our analytics, you can have a big effect on the network. And that's something that was observed in a number of types of networks in the early 2000s. So now to get into the computational aspects that we do. So 3 stages to our in-silico piece. First stage is network model construction. We formulate this as a biological inverse problem. We have a bunch of measurements shown there on the left-hand side, and so we take cells, healthy versus diseased cells, for example, and make measurements, or you can take cell lines, which are exemplar tissue that has been sort of kept in the lab, and do other types of measurements on them. But what you end up with are a lot of different measurements from, for example, a cancer versus non-cancer cell, and you can measure the state of every protein in the cell, the state of every gene in the cell, the state of the copy number variation of every gene in the cell. Whole host of different measurements you can take on different molecules in the cell. And what we want to do is generate a model of the thing that caused those measurements in the first place. So that is a typical inverse problem formulated statistically. And there's a number of ways of solving this in our field. I guess the general class of solution are called active module detections. The most typical approach used are heuristic, greedy search approaches. We typically use a more exact approach. Someone actually did the mathematical proof and said there's an exact solution using what's called Steiner trees. I'm not going to get into the maths of this. There's 2 different illustrations here. The key is that with the Steiner tree approach, you can actually add weights to the network's nodes and the edges that come from those biological measurements. And then use, in fact, optimization approaches. They are, we typically will formulate it as an integer programming problem and solve it using optimization approaches. We use IBM Cplex optimizer. We found it's one of the better ones. I'm not going to go into this much more. It doesn't involve the sort of things we're talking about today, but that's sort of definitely one of our computational aspects. Couple of points on the bottom there of future challenges, which are basically the data's getting bigger, and that's going to affect more or less everything we do. The second computational piece is involved with the compound mapping stage, the final stage, and this is a machine learning a problem. So the bottom right there sort of illustrates in these matrices what we're trying to do. The empirical measured in the lab data for this is very sparse. What the matrix represents is a collection of compounds. We have about 15 million of those in our database at the moment, against the activity of every protein in the human proteome, which is about 22,000 at the moment. The vast majority of those data points haven't been measured in the lab, so we use the data, the ones that have been measured, and use machine learning to predict the ones that haven't been measured. On top of that, we also use we use a combination of approach using model ensembling, typical approach that's used in machine learning these days, and we ensemble approaches that just use the structure of the data, so matrix completion approaches, with other approaches that utilize features from the compounds and/or the proteins. So some requirements on this. This data is very heterogeneous. I don't know if there's any people who work in machine learning in the audience, but if you've got massive data sets, they're hard to work with. Things are slow, so you typically will subsample your data sets and build your models based on that smaller set. Very typical approach. You work on something small, build it up until the final set. What we've found, because of the heterogeneousness of this data, is that the results we find on the small sample sets don't scale to the bigger sets. They're extremely hard to come up with a sample data set that represents your full data set. So we've stopped doing it. What we wanted is a system where we could actually just load all of the memory into data at once and operate on it. Some numbers there. I just did a back-of-the-envelope calculation. It's about 1,200 gigs to load the whole data set, our current data set, and that's getting bigger all the time. With a float, we can do some sensible data filtering, get that down to about 300 gigs. So solution we used, we actually use H2O for this, competitor of Spark, I guess, some level, but it's basically an in-memory distributed machine learning platform. Reason we chose it, well, one, it did what we needed to do. We could get it all of the data into memory on our grid, but then I'm guessing Spark could have done the same thing. It had the algorithms that we wanted to use already implemented in it, so we could focus on our business problem rather than implementing machine learning algorithms. But the critical thing, against Spark at least, was that our data scientists preferred it as an environment to work with. It seemed much more set up to support the needs of the sort of working data scientists rather than over Spark. So now the network analysis piece. I'm focusing, there's more slides on this because this is where we utilize the grid gain solution. So I mentioned the videos didn't play, but what they were trying to show here is what happens to a network when you randomly perturb it, errors in this case, or you attack it, you go after things that are specific nodes. And that's illustrated here. This is a recreation of a graph, as I mentioned, that was published in the early 2000s, actually in 2000. So what you see in orange here is what happens to a network when you remove nodes randomly. So the x-axis is how many nodes have been removed from a network. The y-axis is a measure of the change on a gross network structure. In this case, it's the average shortest path, as mentioned across the top. So if you look at, what's the path between two nodes, and then you look at that across every pair of nodes in the network and take the average. What you can see in orange is that you can remove quite a dramatically large number of nodes and have very little effect on the ability for nodes to communicate within that network. As I mentioned, that is a very common property of biological networks, but also transport networks, the internet, etc. That's why the internet is not susceptible to point failures necessarily. However, if you take out specific nodes, what you see is what you see in blue. You can have a much bigger effect than random. However, they're not that easy to find, those combinations, and that's what our analytics tries to do. So we have 3 core analysis routines that we utilize, and that I'm going to talk about in more detail. They can all be formulated as embarrassingly parallel problems, hence the use of the grid gain compute grid. We have perturbation analysis, which basically generates that graph that I showed you in the previous thing, and it's used effectively as a calibration tool to calibrate the other analyses we do, because the behavior of a specific network, while they all have that general trend, each specific network is different in its specifics. And then we have two other analyses we do. One we call impact maximization. So that is, find me a set of nodes, proteins, that maximize the disruption of the network. It's usually a set of a fixed size, say 10 nodes, 20 nodes. So that's an optimization problem. And then we have something we call compound impact ranking. And as it sounds, we just go through all of the compounds in our database and ask the question, what is the effect of that compound on the network structure? Does it have no effect? Does it have a big effect? And we rank them. This is what we use the grid gains compute grid for. I think more or less every other talk I've seen today is focused on the in-memory grid. We actually don't use the in-memory piece. We came at this a number of years ago, actually. We started this about 6 years ago, and this is what we're after. At the time, Hadoop, I guess, was the standard for MapReduce type algorithms, which is effectively this is what is, but the Hadoop's focused on massive data sets. We're not in that realm at all. It's also focused on batch processing. We wanted sort of online user-triggered processing, and hence our, why we went to the grid gain compute grid. So hopefully I'm not teaching you all to suck eggs here, but what does the compute grid do? It's basically a distributed fork join, or a simple map reduce, as it's been called. So the idea there, if you come across a fork join in Java or any other languages, where you take a task, you split it up into chunks, they all process on different threads, and then you bring it back together again. It's that concept extended to multiple machines, multiple JVMs. We had the additional complication where we had the requirement to run multiple concurrent analysis from different users as well. So, perturbation analysis. This is effectively the runs to generate the data I showed you earlier. How does that map to a fork reduce? So it's statistical in nature, so we repeat it numbers of times for a specific network. There's a random element. That's how many fork joins there are, how many repeats you do. Typically, we'll do 100, so there's 100 fork joins that go on. Each fork will split basically the whole compute job up into a number of tasks, into a number of jobs. Each job basically says what happens to a network when you remove 5 nodes, 10 nodes, etc. And so the whole population of jobs will give you one line along the data along here. When you do the statistics across all of the repeats, you get the error bars. The impact maximization, I mentioned, what's the goal here is to find the number of protein set that maximizes the impact on a network, the change in average shortest path, for example. Exhaustive approach, I actually did the numbers there. So, a typical project will have a network of, say, 1,000 nodes, and we want to find a node set of 20. Number of combinations there is astronomical. You can't do an exhaustive search. So, stochastic approximations, meta-heuristics are typical tools to solve these problems. It basically allows you to explore solution space with some twists to allow you to find global maxima. If you tried to do a hill climb or whatever, you would end up in a local maxima. We use genetic algorithms. It's one specific type of a meta-heuristic or a stochastic approximation, and what's important, I guess, for this thing is that the algorithm is population-based, so it maps very well to a fork join distributed embarrassing parallel scheme. And the algorithm is very loosely based on natural selection. I'll go this slightly in the next slide. So again, how does this map to a fork join? Over time, I guess the number of fork joins is determined by asymptotic convergence. Basically, it keeps going until we get no change or little change. One compute task deals with a generation of solutions. So at every fork join, what happens is that the master node will generate a population of solutions, basically 20 nodes that it wants to test. Initially, that population is generated randomly. As the algorithm proceeds, the population is generated by what's called breeding, combining, basically, the best solutions from the previous generation. That is the basis of a genetic algorithm. And the details of the combining is what maps very loosely to selection and natural selection. So in terms of the fork join, each task calculate everything for a population. Each specific job within that task is the calculation of one member of that population. So some implementation lessons came across. I was asked to talk about things we learned. When I went through these, I was like, well, these are all self-evident, but I guess they weren't because we got bitten by them when we first did it. So they're self-evident now. So our first implementation, which I've called naive, basically for both of these previous tasks, the master node would generate the population of networks with nodes removed that it wanted to test, and then distribute those networks to the worker nodes to actually do the calculations. When we did this, what we found was the parallel distributed implementation was slower than the serial implementation we were trying to improve. And the cost of data distribution just swamped all the CPU gains we got. So we went back to the drawing board. Our current solution, slightly different, but functionally is the same. We basically distribute the network to be perturbed or to be manipulated once at the beginning of the whole run, and it's held in a cache on each worker node. And then what the master node generates at each iteration, rather than generating perturbed networks, it generates a bit vector of nodes to remove. Obviously much smaller than the whole network, and we get the speed ups we would like. One side effect of this, which basically sort of pushed us in a certain way for implementation, was that that network that is now distributed to all of the worker nodes is now shared between multiple worker nodes and multiple threads. So you need to take that into account. We have an immutable data structure for the network that the perturbed networks are created effectively by creating copies of that immutable one rather than removing nodes from the first one. So you've got to make sure that that copy with nodes removed function is actually fast. A lot of work went into making that fast. Otherwise, again, you get that swamps any gain you get if you do it naively. So here's a graph on scalability, and a couple of points I wanted to bring out after this. So what I did here was that we have a pretty small grid, actually, internally. It's got 4 physical machines, 24 cores on each machine. So I just went through and ran the same job with 1 machine turned on, 2 machines, 3 machines, 4 machines, and showed what happened to the speeds. And these are all shown as ratios of the speed. So the orange line, which you can barely see, is the idealized, everything scales perfectly, linear scalability that, of course, you can never get. The blue line is the genetic algorithm. That's pretty damn close to perfect scalability. The other line, however, is the perturbation analysis, and that's quite away from ideal. So why is that the case? And it's basically the issue of job homogeneity or heterogeneity. So again, self-evident now, we've hit it, but it confused us why. But basically, the fastest time a task can take is the time of the slowest job within that task. Obvious, really. But genetic algorithm, all of the tasks are basically removing the same number of nodes from the network, so they're roughly the same computational overhead. They've got about the same number of paths to iterate over within, to calculate average path length, for example. Perturbation analysis, that isn't the case. Some are removing 2 nodes, some are removing 300 nodes, and so the heterogeneity of that job population is much bigger, and that's what leads to that scalability difference that you saw. And the other thing we found is that it's actually, we tried to tune this, of course, and what we found is just a whack-a-mole problem. You tune it for one sort of type of graph, and great scalability. You put another type in, one that's bigger, different set of parameters from the user, goes to hell. So it's actually, we found it really hard. We're in a we can live with it situation at the moment. We would like to look at, for example, the job stealing SPI that exists in grid games, see if that could help. That's normally for heterogeneous hardware. Maybe it can help us with heterogeneous tasks, I'm not sure. Compound impact ranking. This is the final one I mentioned. As I mentioned, the goal here is to evaluate the impact of every compound in our database. Result of one of those graphs is shown down here. I mentioned we've got 15 million compounds in our database. This graph only goes up to 25,000. It's another one that you can illustrate the vast majority of things don't do anything. You can see the vast majority of compounds do very little to the network. That's what's graphed on the y-axis. However, a small collection actually do something quite significant to the network. Those are the ones we're interested in. Those are actually the ones we go on and test and could become potential drugs. So quite simple here. The total, total run across all of the fork joins is basically go through the whole database. Each compute task basically takes a page out of that database we paginate across and evaluates all of the compounds within that page, it's a parameter to tune, and then each job within that task actually takes a sub section of that and sub-page within the bigger page. And that set size is something we tuned. It's actually sort of dependent on hardware knowledge, but our grid's quite fixed at the moment, so that we can get away with that. What we found with the scalability here was something slightly different. It actually depended on network size. And what you can see here is the scalability graphs shown for 3 different networks. The one on the bottom there is for a small network, medium, large network. So large networks scale okay, although not as close to perfect as the genetic algorithms. The small networks, which we analyze a lot of, don't scale at all. Terrible. Again, that doesn't come out very well. I hope you can all see this. It's another way of saying this. Basically, I looked at CPU use on one of the grid nodes while these algorithms were going on. And what you can see in the top there is what happens with a small network. To its right is what happens with a medium network. The one in the middle is what happens with a large network. And the one that happens on the bottom is for the genetic algorithm. And what you can see is that the small one illustrates it very well. CPUs are just not being used. The I/O to the network, to the database is dominating there. It depends on network size, and as I said, for small networks, the compute time is completely swamped by database access time. It's less so for larger networks. We expected that the calculations going into this would actually dominate the network. So that's, I don't know how many times I've learnt this lesson in my career. Measure, don't assume. We're all taught that, and we forget it sometimes. Again, this comes down to another very tricky sort of whack-a-mole problem to tune. There's a lot of parameters that are both given by the user, that depend on the hardware of the grid, and you can tune for one, and the performance of others goes to hell. Obviously, the thing we want to do, and we haven't done that we do want to do, is to investigate the in-memory data grid. When we first set this solution up, it was before Ignite existed. We were paying for grid gain. It was only a pay-for system, and frankly, we couldn't afford the licenses for the in-memory data grid. We're a small company. We obviously can now with Ignite, and so that's something we want to look at today. This week. I'll get it right the 3rd time this year. So just to summarize, so we've basically implemented now 3 different business processes in production for over 4 years. What are some of the technical advantages of the solution? The key one for us, where basically computational biologists, people with disease background, it removes the need to implement parallel distributed processing infrastructure. We basically couldn't have done this solution without something to build on. We just wouldn't have had the expertise all the time to do it. Well, I've said that in the second stage. It facilitates our development focus to be on the business problem, which I guess is what everyone wants, really. Some lessons to be learned. You basically can't forget about Sun's fallacies of distributed computing, even if, which they do with grid gain, it gives you a nice, powerful, easy-to-use API, but you can still burn yourself by using it. As I mentioned, you need to minimize data transfer. These are lessons we found, at least. You need to be aware of how the various parameters of your analysis affect job heterogeneity or homogeneity, and database access can affect even very CPU-intensive jobs. Again, I look at them now, they all seem very self-evident to me, but we did get burnt by them. Business advantages of the solution. So one of the goals we had when we were developing our software platform was to remove as many parts of the we have a bigger process when we go into the lab, and we wanted to move from that whole process all the bottlenecks due to computation that we could. When we started, it was actually, it was quite a big bottleneck on the order of the similar time that we spent in the lab for certain things. It's disappeared now. It's negligible compared to the time we spend in the lab. So, that has definitely been a plus. We can go from concept to start testing something in an idea in a lab in less than 6 months, and most of that time is the intellectual time of thinking about what to do up front. It's a garbage in, garbage out problem. You need to think about the disease and the mechanisms up front, the more time that those biologists have to do that and not worry about the computation, the better the output's going to be. It's also changed the working model from a batch-driven to a real-time exploratory approach. The analogy I like to use here is one of the way that we used to compile code. You'd make a change, press go, and go and get a coffee and come back however long later and get your results. Now it happens in the background in your IDE. It happens in real time, and it's, at least for me, it changed the way I write code. And this is the same thing here. We've basically gone and changed the way that the biologists work because they can sit and explore ideas now much more easily than they could before, explore more hypotheses. It's much harder to quantify the change here than we've reduced 4 months compute time to nothing, but I think it's very real. However, it still can be improved, as you saw in the last one. And the other thing is that quite often you have a choice of algorithms to use. For example, with the optimization one, genetic algorithms aren't the only way to approach stochastic optimization. Simulated annealing, for example, is one of the other big ones. That's not population-based. That's harder to map to a embarrassingly parallel solution. So at some level, the algorithm that you choose can be driven by the ease to map to the underlying, sort of reverses the process. You're choosing the algorithm based on the solution rather than the other way around. But that can actually help. So future, we're still on a very old version of Grid Game, 4+ years old. It's worked, never had a need to change it, frankly. However, I did mention we want to start looking into the data grid things to improve the speed of that one particular algorithm and possibly the job stealing stuff. So we are planning to move to Ignite. The other thing that we're interested in looking, because we've reduced the computational down to nothing, the use of the computational piece now is very spiky. We don't have that many parallel projects going at the same time. So a lot of the time the grid's sat there doing nothing. But when we need it, it would be good to have a much bigger grid. So, obvious there is a, on-demand compute infrastructure. We're thinking about investigating that in combination with a general platform migration to Kubernetes. And I've gone way quicker than I should have done. But I've some slides I didn't think I would get to, but I'll do them anyway because I've got 5 minutes left. This is something that much more academic project that we've been working on. I say we. I've been involved with, but all of the work has really gone on in the academic centers on another approach to fast network processing. And this is not using memory at all, basically. So the idea here, there's a couple of academic projects, Fantasy and Poets. One, Fantasy, it was a small sort of pilot project to go into a larger, a part of the larger Poets project. So these are in collaboration with the Microsystems Research Group at Newcastle University. And the Poets project is actually a cross-university project that involves Newcastle, Southampton, Imperial here in London, and Cambridge. The general idea here is to investigate compute architectures consisting of extremely large numbers of small cores, and we're talking millions. The Fantasy one utilized off-the-shelf FPGA hardware to basically look at, could we take a network I've described and map it to the hardware on the FPGA and use that to speed up network computing? And of course, I wouldn't be saying this if the answer wasn't yes. So the idea here is shortest path analysis. So is that blue coming out or green? So if you inject a pulse at one node, so the idea of shortest path, you start at one source node and you want to know how many hops you need to go to every other node in the network. If you've got the network in hardware, you can inject a pulse into that source node, and at each clock pulse, you get to the next boundary of how far the network can go. Now, one of the properties of these biological networks is they have what's called a small world property. You might have heard this, 6 degrees of freedom thing with Kevin Bacon or whatever. That is the small world property of complex networks. On average, the longest, shortest path in some of these networks is 5 or 6. So if it's 6 clock pulses will get you all of the shortest paths from one source node to there. So he used 7. So 7 clock pulses will give you that. You know, clock speeds in these FPGAs are a lot smaller than in a lot slower than in a regular CPU, but you know, even at 1,000 megahertz, or is it 100 megahertz? Yeah, 100 megahertz, 70 nanoseconds, which, as my collaborator said, you know, this is roughly the same time it takes a conventional machine to access memory. So we can do the whole calculation of one source node to everywhere else in the grid in that amount of time. As I said, this was a pilot project. It was successfully implemented in off-the-shelf FPGAs. We got over 3 orders of magnitude speed up. For example, for a network of 3,500 nodes, which actually was the sort of upper limit size-wise, the speed up over the sort of test CPU-based algorithm was 2,500. The network size, as I said, 3,500 was about the maximum that they managed, is limited both by the FPGA hardware, but actually more critically by the layout algorithms that are used on there. You know, it's a optimization convergence algorithm for bigger networks than 3,500, and I should say that the number of edges in these networks is on the order of 1% to 2% populated. Basically, the layout algorithms don't converge, so you either need bigger hardware systems or better convergence algorithms for the layout, and they're looking at both. And then the POETS project, which is underway, the idea there is to network together multiple FPGAs to give out potentially millions of cores. And that's all I wanted to say. Thanks.